Good morning and welcome everybody to the session titled Biological Basis of Addiction, Research Updates from the Jackson Laboratory. My name is Michael McKernan. I'm the Senior Director for Government Affairs and Community Relations at JAX, based in Bar Harbor. Remembering human stories is one of the important reasons why we participate in this summit and have now for several years in a row. We're really grateful for that opportunity. In most settings and most scientific meetings, there's usually a lot of scientists in the room, and there's sort of the token person from the community, the advocate. You know, there are small groups, but usually the scales are very heavily tilted towards the scientists. It's a very familiar space for us. This really turns the tables on that. This gives us an opportunity as researchers, scientists, to hear directly from members of the community, from you all, to hear stories. And it's incredibly important and I think invigorating and centering for both myself and my colleagues. So I think that's a kind of a good preface to start. Jackson Laboratory, we are one of the largest employers in Eastern Maine. We're a very unique institution where we're sort of three things in one. We're a basic research institution, biomedical research institution with about 60 professors across a couple different institutes with hundreds of staff, including scientific technicians and specialists and a lot of graduate students and trainees. We're also an educational institution. We're very proud, for example, of several doctoral programs that we have, including in partnership with the University of Maine, with the University of Connecticut, and with Tufts University. And we're also very well known for our mice. We produce, we design, we distribute specialized tools, mouse models, for a range of human diseases. And these tools are used worldwide by scientists trying to investigate the origins of cancer, Alzheimer's, of course, addiction, substance use disorder, and many, many others. So that's kind of what we do at the Jackson Laboratory. So all this work is supported by nearly 3,000 colleagues worldwide and 1,500 employed in good jobs here in the state of Maine. So we've assembled an expert panel here today to discuss the biological basis of substance use disorder, including the genetics of addiction, overdose, and withdrawal. And while broad understanding of the biology of substance use disorder is critical, research is increasingly focused on personal genetics. How my genetic background, your genetic background, when combined with environment and other factors, provide important context for understanding how this disease manifests differently and may be treated differently in different patients. So as a panel, we know this is a very heterogeneous audience, including there are some other researchers in the room. There's certainly some physicians and clinicians in the room with deep knowledge of these biological topics. There are individuals with lived experience and individuals who have last encountered the word genetics in high school biology or maybe on YouTube, which is fine. We all share a goal to understand the opioid crisis from many angles, including through scientific research. So please ask any question, don't be shy. We don't have a mechanism in this room to come to a microphone and ask a question, so we do ask you to use our Slido mechanism. So log into Slido or take a picture of the QR code and type any question that you want, and then when we get to Q&A, I will moderate those questions. So what we'll do is we'll invite each speaker to come to the podium and talk about their research interests for about 15 minutes, and then we'll do Q&A at the end. I think we'll now get underway. Let me just do a quick introduction of our panel. So our first speaker will be Alyssa Chesler. Alyssa is a professor in the JAX Center for Addiction Biology and is Senior Director of Integrative Data Sciences based in Bar Harbor. Our second speaker in the middle is Jason Boubier. Jason is a senior research scientist, also in the Center for Addiction Biology at JAX in Bar Harbor. And our final speaker on the end, Jake Byerly. Jake is a postdoctoral associate in the laboratory of Vivek Kumar, also in the Center for Addiction Biology based in Bar Harbor. So this is a fantastic panel, and we'll get underway with Alyssa.
Thank you, Mike. Thank you for the introduction, and good morning, everyone. Happy to be here. What I wanted to talk today about was something that we've been learning over the decades of biology of addiction research, which is more about how we come to understand the nature of the role of genetics and variation in the biology of addiction. Substance use disorders are among the most complex of what we call complex diseases. When we say a complex disease, we mean multiple genes, multiple environmental factors, life experiences, development, all coming together and interacting to produce the outcome that is a disease, a disorder, a trait, a behavioral state. Many, many effects coming together. Human genetics has revealed, at this point, hundreds, if not thousands of variants, differences in each of our DNA that are associated with our risk for these conditions and how they play out in our lives. And what we learned from mice and other models is different ways that we can understand, study, and characterize this complexity so that we can learn about those individual differences affecting each person in their own way. This is a graphic of all the roads that lead to Rome. And when we think about an individual with a substance use disorder, we can look at this and say everyone got there on their own journey. And that's true from the adverse life experiences, maybe great life experience, and a lot of biological predisposition, what drugs were available, when did you start using those drugs, how did those drugs interact with each other, and how did you respond to the effects of those drugs? All of these things come together in many unique paths, bringing us into the place that we are today. So it's not just one major genetic variant. Geneticists used to look for the smoking gun, but it's many thousands. And so here we have the first genome sequenced, trying to figure out what's happening here, and one of the biologists says, I think I found a corner piece. We know that only some people who use drugs develop a substance use disorder, and that role of the genetics is different for different drugs. So we can see that given exposure to a drug, nicotine, very highly addictive, alcohol, 23% of people using alcohol develop an alcohol use disorder, but opioids, cannabis, we all know these are addictive as well. And then if we look at a pair of twins, even those raised apart, there's a probability that if one twin develops a substance use disorder, the other one does as well. And we can see that even that varies differently for different drugs. So cocaine use disorders are very highly heritable. So one twin gets cocaine use disorder, the other twin may have it as well if they're both exposed to cocaine. Opioids, quite similarly. Even smoking, gambling, somewhat highly heritable. And the hallucinogens and other drugs are less highly heritable, but still to some extent, twins might share that. That's saying to us that there's a great role of biology and shared genetics in driving whether somebody is exposed to those drugs, develops that substance use disorder. So really, when we think about addiction biology, we're talking about many roads to many Romes, even the one here in Maine. What we learned from mouse genetics, we can look at each of these factors in isolation, in combination, and examine what biological variation is associated with which of these different traits. And we use techniques we call systems genetics to find the genes and variants associated with many, many of these traits. And we look at data from lots of different sources, including human genetic studies, mouse genetic studies, and other organisms in concert to try to best understand how we can characterize what's known from human populations. Now, historically, a lot of the behavior studies that are done in laboratory mice were done on a very few mouse strains, some very famous mouse strains, but there's a whole world of mouse strains out there, not everyone's famous. And when we look at all of those strains together, we see that each of them differ in different ways with respect to behaviors that look like things that we see in people with substance use disorders. How much do they explore their environment around them? How much reactivity do they have to certain stimuli and situations? Do they seek out novel environments? Do they prefer to hunker down and hide in the dark? What's their circadian cycle? Many people develop a substance use disorder in the art of managing an abnormal circadian rhythm. And then we see also different patterns in these different strains of mice, and how much drug they will self-administer, at what rate do they acquire that self-administration, and if the drug is taken away, do they keep seeking it, or is it easy for them to stop seeking that drug? And so each of these measures, each of these colored lines here represents males and females from a different strain of mouse, and we can see that they differ in different ways. And when we can actually shuffle the genetic deck, we take these 8 strains of mice, breed them together in different ways, and we can then use those populations that come from that shuffling of that genetic material to connect specific genetic differences with specific behaviors and specific biological characteristics. Genes encode for proteins that might be receptors for neurotransmitters. They might be chemicals circulating that are manufactured by enzymes. All of these tiny differences affect the biology of the individuals. And we also know that there are places in the genome where an environmental event might alter to what extent other genes are expressed. So those life experiences change the patterns that our DNA uses. It's epigenetics. They change the patterns of gene expression throughout the brain and throughout the body. Now, we can actually bring together data from mice and humans in different ways, and when we do that, we can see that each of these strains of mice resembles people differently. And so when we look at genes expressed in people who had chronic cocaine use, we can see different strains of mice match to that pattern of people with chronic cocaine use in different ways. So different strains resemble the individuals, and some of them resemble them much better than others. We also see that in different strains of mice, there's a different risk for the lasting biological changes that occur in addiction circuitry. So here we're looking at 2 strains of mice. Both of them acquire cocaine self-administrations. We don't say the mice are using cocaine. We don't know if they're using it or not, but certainly they will learn to press a lever to take cocaine, and both strains will learn to do this. But if we turn off the cocaine, remove the drug, one strain of mouse restores the same pattern of gene expression that it had prior to exposure to the drug. But the other strain of mouse doesn't stop looking for the drug. And in that strain of mice, the genes that came up during learning the self-administration of the drug don't come back down. And so one of the things we might look for in terms of developing therapeutics is to say, is there something that we can do, even meditation, right? Even a behavioral intervention, even nutritional intervention, is there something that we can do to make the brain state resemble the state of the brain that can stop using the drug? When we look across mouse strains and many different behaviors, we can find the relationships among those behaviors. So we can ask the question, is preferring alcohol to water, is that correlated better with stress reactivity? Is it correlated with behavioral despair? Do mice that seek cocaine, are they more impulsive? Are they high novelty seeking? These are certainly things that we might speculate, but we can actually test which of these behaviors are related to one another. And when we look at a large population of mice, and we look at the behaviors that are associated with learning cocaine self-administration, extinguishing it, stop looking for the drug, and reinstating it, going back to seeking that drug in the presence of cues. In our case, some people look at stress. How does stress turn back on drug self-administration? We look at these different behaviors and we say, what background behaviors are also associated? And it turns out when we look at behaviors associated with impulsivity, we see that those are associated more with starting use and to some extent not extinguishing the use. But as we look at extinction and reinstatement, a relapse type behavior, but starting to seek the drug when you're faced with cues associated with the drug use, those are associated more with anxiety. It was previously believed that in mice, and certainly in people, that it was those novelty seekers that are going out and looking for more drug, taking more drug, increasing their use. But there's also a component here that the anxiety potentially associated with not having the drug available anymore is an issue that for some individuals can make it more difficult to stop using. We can connect each of these different characteristics to different sets of genes and different biological pathways, and there we can start asking about questions about tailoring our approaches to individuals that have these different behaviors driving their substance use disorders. It's a long road from mouse genetics to these types of interventions, but we're on that road. Here we looked at human genetic studies of opioid use disorder, and we can see again the same point. Many of the genes associated with opioid use disorder in people are associated with lots of different biological and behavioral characteristics of mice. So it's not a one-size-fits-all solution. So when we get these thousands of genes associated with a human disease, we need to go back in and say, what is it that each of them do? And which of them are important to us? Which of them help us predict and treat that condition? We can use these data to actually identify which mouse strains best match any type of individual with the disorder. And so here we've done that. We've actually gone through the mouse populations. We did some data analyses that allow us to match those things. And we looked at other data that people had collected historically in different strains of mice. And we can actually see mice that were predicted to have low substance use disorder type characteristics indeed will take less cocaine, respond less to novelty, decrease their exploration in a novel environment, less anxiety. And those that take more cocaine, or rather those that were predicted to more resemble people with the substance use disorders, also exhibit those characteristics. And we can, again, drill down to each individual gene and variant in one of these massive human genetic studies and go back in and use all of the data that's been collected in laboratory mice for nearly a century now and ask questions about what does each of those genes and variant do from the rich information here. So there's a compelling new drug that's in clinical trials around alcohol use disorder. It acts on an enzyme here, it's PDE4B is the word we use to call it, phosphodiesterase 4B. We can ask what that gene does, and when we look at the mouse behaviors across every different drug, there's hundreds of behaviors represented here, we see that that gene is involved mostly in withdrawal-related phenomena. So we say that this therapeutic might work best in alleviating the consequences of withdrawal and in that way reducing the amount of reuse, withdrawal-mediated use. So in summary, there's many, many differences in life experience, drug exposure history, and genetics. They come together in many different ways to shape an individual's addiction trajectory, and that's what we see when we look at the biology. Addiction histories and biology look very different in different individuals, and we can study these in mice and humans to understand, evaluate, and target interventions for addiction. A lot of people involved in all of the work that we do, and I'm especially grateful for the support of not just the Jackson Laboratory, but the NIH, NIDA, the National Institute on Drug Abuse, and the National Institute on Alcohol Abuse and Alcoholism, who have supported a couple of decades of our work on this topic. Thank you very much.
All right, thank you, Alyssa, for that excellent introduction to our topic and how overarching the Center for Addiction Biology at the Jackson Lab is covering, from one end to the other of addiction. I'm going to be focused on right now the risk associated with overdose. So overdose deaths, actually, we've made progress in the last few years. We've had decreases in overdoses because of the Narcan that's been distributed by many of these tables out here and many of these organizations. So thank you. This is a major success for human health and for public health in general. Opioids. So we know that they're great. If you have a surgery and you come out of surgery, you probably will need an opioid. It's really important for taking care of that pain, for the analgesia, but it also causes respiratory depression, cardiovascular depression, decreases your motility in your gut, constipation. Too much of an opioid causes sedation. If you get the right amount, you can get a euphoric approach as well as a urinary retention. So there's many aspects to the use of opioids. And as we know, there's been a history in the United States recently of an overprescription of opioids with the promise that they're not addictive when in fact they are very highly addictive. What happens during overdose? Opioid overdose generally results in death through what's called opioid-induced respiratory depression. So there's this region of the brain called the medulla. It's kind of in the brainstem area. There's a cluster of cells that contain opioid receptors. They respond to blood concentrations of oxygen and carbon dioxide and determine how fast or slow you should be breathing. Opioids bind to those receptors, and then they kind of mess up with the ability to regulate and respond appropriately. And so your respiratory rate can fall, and eventually death can result. And as I mentioned, Narcan or naloxone has been a great thing to discover, and its deployment in the United States has been amazing. Naloxone reverses overdose by displacing the actual opioid on the receptor. So in the little pink ball here is the opioid, like morphine or heroin. And then the naloxone comes on and kicks it off, and instead binds really tightly to the receptor. And the opioid goes away, and they no longer are experiencing the effect of the opioids. So their respirations come back, the euphoria goes away, withdrawal comes on if you were in chronic use. So it can be not pleasant to be revived by naloxone and Narcan if you've been a chronic user. Naloxone, however, in the advent of the newer synthetic opioids such as fentanyl and carfentanil, naloxone finds a difficulty in competing that opioid for the receptor. Fentanyl was designed to bind to the receptor really strongly, so naloxone finds it difficult to bind to it, and you may need to be re-narcanized. You may need to be repeated doses of Narcan to come through because of the potency of the opioids. All right, this slide is to show us that we are all very different. Notice the minifigs on the left; they're all very unique, but yet we're 99.9% similar. And a lot of these differences we can see at the level of outward appearance: red hair, green clothes, or brown hair, or whatever in these figures. But a lot of things that you can't see are your susceptibility to opioids. Some people, for example, the same dose that you use would actually be 100 times too much for them. Everybody has an individual susceptibility, and you can't see that by looking at their face. Just because they have the witch's hat on doesn't mean they're going to be super resistant, or the Batman cross mean they're going to be helmet mean they're super sensitive. Now, we can't really do these experiments that I'm going to do about overdose sensitivity in humans. Instead, we're looking at mice. And mice, while not identical to humans, are 92% identical. So they have 92% similarity in genes. A lot of their arrangement is the same. The same region of same chunk of genes are arranged in the same order on a chromosome, a different number in mice, but it's the same arrangement in humans. So mice are a great tool. And just like the human population is very diverse, on the left here you can see we have a diverse mouse population that Alyssa showed us earlier called the diversity outbred population, where all the mice look very different. Their coat colors vary. Some are really cute. Some of them are all one color. Sometimes they have spots on them. This mouse population to each other is 99.9% similar, but there are differences besides the coat color. And if you look at the top two mice, those mice need to have a higher dose of opioid in order to have an effect. If you gave them just one capsule, they wouldn't feel it. They need like 100 capsules to feel the effect. Whereas that mouse down on the bottom, those white and black mice, a single dose or single capsule is enough. So that sensitivity, you can't really tell by looking at somebody what it's going to be. But if you look at the arrow pointing to the DNA, there are regions of the DNA that can tell us, that person's probably very susceptible to the use of opioids. They probably shouldn't use them that much, or their dosage needs to be monitored closely. So why are some mice or humans more sensitive than others? So on the top we have an NZO mouse. This is a mouse that one pill would be enough, and actually would be too much. About 50% of the mice of this type given one pill would die. But there's another strain, a black 6 mouse, that needs about 107 or 50 times as much opioid, in this case fentanyl, to have the same amount of population death. So what's happening? If you look in the figure on the left, the number of breaths, that sensitive mouse has a lower breath rate. It gets opioid just like this. So the red and the blue both are going to receive opioid after 15 minutes. You see a depression of respiratory effect, the frequency, and it continues over time. But the blue line starts out higher. It has a higher respiratory rate, and then it drops. But if you look at the volume, the amount that the mice are exhaling, if you look at the depth of the breath in the bottom left, those mice that are most sensitive actually take larger breaths, and then when given fentanyl, actually increase the depth of their breath instead of decreasing the depth of their breath. So you would think they might actually be more resistant, but in fact, it's counterintuitive. Those mice are more sensitive to opioids. And you can see correspondingly the oxygenation of the blood. The blue figure line shows the more resistant strain of mice has higher oxygenation, and then the more sensitive strain, the red, has lower oxygenation associated with the effects of the opioid-induced respiratory depression. So the blood is not getting the oxygen from the breathing. So naloxone and Narcan are great, as I've said, but that re-narcanization, needing repeated doses, can be a problem. So is there another way that we can stimulate breathing through other respiratory control centers within the brain? Can we, stimulate a respiratory uptake in addition to trying to replace the opioid at the receptor? And so that's pushing us towards these other ideas of naloxone plus a respiratory stimulant or an adjuvant. So here the idea is when you're given naloxone, you're also given another compound. And we've identified some of these compounds in our studies with mice using data from humans. We started with, got to mice, identified a compound, and then if we give that compound to you when we give you Narcan, as the Narcan and naloxone wears off, this compound is going to take off and it's going to increase your breathing rate on its own. So it's like a 1-2 punch. The one punch would be the naloxone affecting the opioid receptor, and then the second would be that secondary compound, which is going to help stimulate the respiratory depression. And so this is the type of finding that we've had in the last 5 years of working at the Jackson Laboratory on opioid-induced respiratory depression, is identifying these possible new things that we can test for therapeutic advancement in the clinic to help with these other drugs like nitazines as well that are hitting the street, and carfentanil. So the takeaways, a key thing that I didn't mention, but that is key, is that addiction is a disease. It has a biological basis. It's not a moral failing. It is a chronic brain disease. Opioid overdose is a biological mechanism, and each person has their own risk potential. Genetic variation in a population can help us identify new targets, things that we hadn't thought of to look at for new reversal agents to combat the current and the emerging threats, the different drugs that are coming out. And model organisms give us the ability to try and separate the different functions of the opioids. Is the opioid working at the level of gas exchange? Is it working at the level of respiratory rate? What level of mechanism is the opioid working with the genetic variation? And none of this is done alone. As Alyssa said, there's quite a large team and funding from the NIH that supports us, and a group of lots of collaborators and talented people at JAX who facilitate. Thank you.
All right, I'm going to wrap us up. I'm going to talk to us today about my work at the Jackson Laboratory as a postdoctoral associate in Vivek Kumar's lab, trying to understand the genetic modifiers of opioid withdrawal. And really my work is asking the question, are we studying complex behaviors across genetics in the right way? And how can we improve the models that we use to more appropriately interrogate the things we care about in humans when we go to treat them? So we've talked about this a lot, but opioid use disorder is this compulsive recurring cycle of drug taking, intoxication, withdrawal, and then anticipation and preoccupation with finding more drug. And that's kind of displayed in this very academic and simplified figure that's excluding social and economic and other sort of environmental factors. But the reason I'm really interested in withdrawal is because it provides one of the first barriers to recovery. Withdrawal drives people, patients, to seek more opioids and consume more opioids. And we know that alleviating withdrawal helps patients because there are FDA-approved medications that we can use to blunt the severity of withdrawal and improve patient outcomes. But there are only one or two medications, and they work through the same pathway, the same biological pathways. And as you can see, withdrawal is not mediated by just a single pathway. There's no one single lever that we can push on to change withdrawal. There's many different opportunities. And so I think that means that there are more opportunities that we're not taking advantage of to help people overcome that initial stage of withdrawal and enter treatment and hopefully improve their outcomes. So that's why I study withdrawal in opioid use disorder. Why do I study addiction? Genetics. We know that genetics influences a person's risk to developing opioid addiction. And we know that in a few ways in humans. There are twin studies that Alyssa was talking about that demonstrate that there's heritability by comparing identical twins. And while the estimates vary, it clearly demonstrates that a portion of your risk for developing opioid use disorder is attributable to your genetics. And then we can also take an approach called the genome-wide association study, where we take lots and lots of people, like 23andMe data, and we see what specific changes in a DNA sequence across a lot of people put you at more risk for opioid use disorder. And by doing this, we've demonstrated specific genetic regions and genetic variants that we think are specifically contributing. And the hope is, right, that if we understand the genetics of addiction, it'll help us understand the biology of addiction. And by understanding the biology of addiction, we can design new treatments and therapeutics to help treat opioid addiction. Okay, so that's why I study withdrawal. That's why we study genetics in the context of opioid use disorder and withdrawal. So why mice? And it's because mice are excellent models of addiction. I'm using some loose language here, but mice exhibit a lot of the same behaviors that humans do when they're exposed to opioids, right? Mice work for opioids. They develop tolerance to opioids. They experience withdrawal when they stop receiving opioids. And that face validity really helps us be confident that mice are good models to study opioid addiction. Additionally, mice share a lot of the same anatomy, the neuroanatomy that mediates addiction in humans, meaning that when we find mechanisms in mice, we can be confident that there is probably an analog neuroanatomically in humans. And then mice also allow us to model this complex emergent phenomena that is addiction, right? So this is something that would be very hard to study in cells in a dish or using a simulation, because there's a lot of factors, like Alyssa discussed earlier, that contribute to the development and the sustainment of opioid use disorder and addiction-like behaviors. And then finally, we know that genetics, like humans, has an impact on addiction behaviors in mice, and that genetic variation modifies that sort of response to opioids. So with all of that, let's talk really briefly about how we study addiction through genetics in mice. And I think we largely do it in two ways. We can take advantage of the natural variation in genetics across mice. So we have these strains of mice that are represented by these little bars. And within each strain, right, within the AJs, for example, the mice are genetically identical. But across strains, they're genetically distinct. Right? And so we can compare the way that those mice respond to opioids and then work backwards to try to understand how the genetics are mediating that change. We can also go within a single strain, and we can take a specific genetic element and we can use genetic engineering to remove it or delete it or change it, and we can see how that specific strain or that specific change on an otherwise genetically identical genetic background changes opioid responses. So that leads us to kind of this sort of central workflow that we've got, this central hypothesis where genetic variation is leading to some dysfunction within a cell that changes the way the cell behaves. That cell changes the way that its neighbors behave. And from that sort of circuit or brain-wide dysregulation, it changes the psychological experience an animal, which is to say a human or a mouse experiences, and that changes behavior of the animal, be it a mouse or a human, right? And so what we try to do is link these genetic variants to changes in behavior and then work inward to this point where we think novel therapeutics live, right? These specific proteins or transcripts that we're looking at. And with things like self-administration, that's really nice because the behavior that we're looking at, we trust to model the behavior that we care about in humans, which is how much are they willing to work or go out and seek a drug. But withdrawal is trickier because withdrawal is sitting in this altered latent state, this altered emotional state. We care about how unpleasant opioid withdrawal is, and we predict how unpleasant that is with how the mouse behaves. Because unlike humans, we can't ask them to self-report their discomfort. And so if we try to study opioid withdrawal across genetics, we run into this fundamental problem, which is this arrow, is this relationship between discomfort and behavior the same across genetic diversity, or is it dynamic? And if it is dynamic, how do we compare across genetic diversity and actually start to understand how genetic variants affect this sort of discomfort state? So the way we traditionally do this in mice is we use single measures. We put mice in these mazes and we see how much time they spend in an exposed or enclosed arm, or we put them in an arena and we see how much they jump. We record single behaviors. There's other ways, but these are sort of examples. And I think you can intuit that this is inherently a limiting approach. It's like looking through a keyhole and trying to see what's in the room beyond it. You've only got a certain amount of view of resolution into, again, that discomfort that you're interested in opioid withdrawal. And this is not how we do it in humans, right? We use these clinical opioid withdrawal scores, cows, and there's 11 items. And I think you can intuit that if you just limited yourself to one item, you might be able to choose a good item to predict somebody's withdrawal, like anxiety or perhaps restlessness, but you're still inherently limiting the amount of information you can understand by going from 11 to 1. So the way that we try to overcome this in the lab that I work in is we use these sophisticated computer vision AI approaches to get more information about how a mouse behaves. So we put an animal who's experiencing withdrawal or not in this arena, and we allow it to move. And as we allow it to sort of voluntarily express its behavior, we overlay these points on the mouse. And by understanding how these points change over time and in relationship to each other, we can infer what the mouse is doing. And by doing this, we can go from simple measures like distance traveled and how much time it spends in the edges to a really rich index of behaviors that more appropriately captures the entirety of the mouse's sort of expressed behavior in response to withdrawal. So the central question then is how does opioid withdrawal differ across genetics if it does differ across genetics? And to answer that question by applying those techniques, we're going to use these star players. We're going to use these genetically diverse 9 strains of mice that are, again, identical to each other within a strain, but diverse across a strain. We're going to have them experience opioid withdrawal, and we're going to see how their behavior changes. And this is a busy slide, but what I want you to know is each bar is a strain of mice. And the bar that goes above 0 means it increases in response to withdrawal, and a bar that goes below 0 means it decreases in response to withdrawal. And I think you can kind of take away from this slide that if you chose one of these behaviors and you ordered the severity of withdrawal in these mice based on those behaviors, you would end up with 6 different orders. And that's fundamentally the problem that I think we're facing. So the fact that they're different doesn't necessarily mean that we can detect withdrawal. That's another thing that we're really interested in. So if we take all of these behaviors and we do a statistical test and we kind of cram all 600 features that I get into an XY plane, right? We would hope that we would find all of the withdrawal mice in one area and all of the naive mice in the other, right? And that would help us be confident that we can identify withdrawal across genetic diversity, even if it looks different across genetic diversity. And that's exactly what we see here. So this is a little overwhelming perhaps, but the red dots are mice that are experiencing withdrawal for each of these 9 strains, and the blue ones are mice that are not. They're just at baseline. They've been administered a control substance. And you can see in all 9 of these, we get a group of red dots and a group of blue dots, which gives us confidence that we can actually resolve withdrawal. What's exciting is when we combine all the mice together, we also get this shared component, this shared clustering that seems to work across all of the genetic diversity that we've tested here. Which means that excitingly, there is some shared behavioral component in the way these mice express withdrawal, and that might help us compare withdrawal severity across genetics. So the reason this matters is I think that this tool and these approaches are going to help us move away from single reductionist ways of studying opioid withdrawal that might be missing what's most important to patient populations. And by expanding into this really rich behavioral index, we improve our ability to help patients by better studying withdrawal. We can study that, we can improve the way that we study withdrawal across genetic diversity, like I just said, but we can also build really sensitive ways of testing withdrawal in individual genetic backgrounds and use that to screen novel therapeutics or look at the effects of genetic deletions across a wider selection of genetic diversity, which will give us more confidence again that what we find will work in genetically diverse humans. Yeah, thank you for your attention.
All right, thank you, Jake. Thank you to our other speakers. We now have a few minutes for questions, and I'd again invite you to go to Slido and provide your question there. I'll keep an eye on that window while I have a few other questions that I would like to ask the panel. We'll get us started. So each of you talked about genetic diversity, you talked about individual genetics, individual variation, and I think we're seeing in other, particularly cancer, I think about the Maine Cancer Genomics Initiative, which is another JAX program that really focuses on individual genetics of cancer patients and tying them with clinical trials and treatments that are specific to them. We are seeing a range of understanding of precision medicine. So could one, and Jake, maybe I challenge you to take the first crack at this answer. How does precision medicine, or how do you think precision medicine might begin to inform withdrawal treatments or approaching a withdrawal treatment in an individual patient?
Sure. So I think that there's 2 ways precision medicine can help in this field. And the first is the actual prescription of opioids and what pain management medication is prescribed to patients to reduce the risk of transition to sort of dependence and entering withdrawal. But the other way is, we've talked about how there's genetic variation that puts or predisposes you to addiction risk. You can kind of think of the brain as a bunch of knobs and dials, and as you turn one up, you might predispose, someone to addiction, and understanding where those pressures are within an individual patient may help us treat that patient better. I think to do that, though, we need a bigger toolkit to help treat opioid withdrawal, right? When you only have one medication and it works through the same way, it's very hard to apply precision medicine approaches. And so things that Alyssa and Jason are doing, they're trying to better describe the risk of addiction genetically are really helpful in achieving that sort of precision medicine approach in the future.
Okay, thank you. Jason, moving on to you, I have a question about the naloxone plus respiratory stimulant strategy. I'm curious, how close are we to a next generation reversal approach that utilizes what you just described?
I think that we're closer than we've been at the Jackson Lab. I've actually put in a patent disclosure indicating that I have a compound, and I have like a year to really bolster my support for this disclosure. So I'm really excited that maybe there is a additive that we can add that can help prevent that renarcanization, where people can just get that one shot of naloxone plus or Narcan plus, and it take them through, and they don't need the multiple hits. So I think that in the past 5 or 6 years, thanks to my funding from the NIH, I've been able to actually move my program quite a bit forward.
That's great, very exciting. Looking forward to more reports on that in the future. Okay, some questions have definitely come in from the audience, so I'm going to take a look at these. And this is an interesting one. How do GLP-1 compounds figure into research? Alyssa?
So we haven't done any work yet with GLP-1, but they are showing a lot of great promise, just from people who've been using them for weight loss and type 2 diabetes. Many people report that has reduced their alcohol use and other drugs. So they are the subject of a lot of new research, and I think they're quite promising for some people, right? Everything will work differently for different people. Some of our work we've identified in mice is that there's definitely a biology of responding to the perception of scarcity and satisfying of that need. And so drugs that are working on the circuitry of hunger and satiety might be tapping into the same circuitry. GLP-1 may not be getting at the root cause of that change. We talked a little bit about epigenetics. What is it that switches on when people are faced with a lack of resources in their environment? And in some people, is that switch on naturally at birth? They are basically born in a condition of sensing that they don't have enough resources, or sensing and that they don't have enough, even when they are in an abundant society or in an abundant socioeconomic status. That perception of need feeds the desire to use. So having drugs that work on those types of mechanisms is going to be an important direction in the future for sure.
Great, thank you. Sticking with the question of epigenetics, a question came in. Does anyone have thoughts? And I will connect this back to a prior panel we did several years ago and mention the work of Jennifer Honeycutt from Bowdoin College. So anyway, the question is, are there thoughts on trauma and early life adversity and the role of those things, and how it influences genetics and susceptibility for addiction? Is there a biological basis originating in trauma or early life adversity?
Absolutely. There's a number of different consequences. Chronic unpredictable stress and the number of adverse childhood experiences certainly influences many different things that play into a substance use disorder. So one thing that we see in people with chronic trauma exposure is changes in their executive function, changes in behavioral inhibition, changes in organization, and many things that are necessary skills for successful recovery. So those same executive functions, those frontal lobe functions, are affected by different drugs. So adolescents using cannabis have lasting effects on executive functions that may make it more difficult for them to change their addiction-related behaviors in the future. There's also effects of that chronic stress on stress reactivity, so how those individuals respond to future stressors. And potentially, again, how does stress and threat influence the extent to which individuals are activated to seek resources? And let's say drugs, rewarding drugs are those resources, and that reward may come from the relief of pain, the relief of stress, and the relief of anxiety, or it may be actually a pleasurable stimulus. So all of those life experiences do alter the biology, and they do predispose individuals to other behaviors that may impact either the extent to which they use substances, the extent to which they maintain that use, and then what challenges they might experience throughout their recovery.
Great. Thank you, Alyssa. And again, just to mention Jennifer Honeycutt, Bowdoin College, for more specifically on adverse childhood events. Let's see. So the speaker has alluded to a future where clinicians can personalize care and take into account a predisposition for dependence. What would that look like? The follow-up question, universal genetic testing for everybody or something else? How far are we from that in practice? Jason takes the bait.
All right. I think pharmacogenomics is an emerging field that's been shown to be very effective for certain conditions. For example, warfarin, a blood thinner, you can predict based on your genotype or your genetics how sensitive you're going to be and how frequently you're going to have to do have your blood work done to see how the warfarin is working, or Coumadin is another name for it. How close are we to this? I think psychological medicines really could benefit from this in the future, because there are many paths to Rome. There are many ways that you can have major depressive disorder. If we can find some ways to separate the treatment paradigms, because there are some that are responsive to SSRIs and there are some that are not. If we can associate that pre-existing genetic mutation with an effect that works, or meditation or yoga works for this particular genetic setup, then I think that would really move things forward. How close are we? Yeah, there's lots of issues with privacy and insurance and all those things.
We don't yet know, Alyssa.
Yeah, I would say, and the complexity of the situation. Not to say that it's hopelessly complex, but we are talking about something that's far more complex than individual enzymes and their effects on blood thinners. So it's I'd say we're not there yet. Single genetic tests or even a whole genome isn't going to precisely predict an individual's addiction risk. And so we have to consider all of the information that we have about people. And look, many people know about their own family history, and that's a good indicator of risk. And then the question becomes, what do you do with that family history? Is that family history confining, or is that family history suggesting a different set of behaviors? And even knowing that family history, is it even something for some individuals that they can willfully overcome, or is it just something that might bring them closer to an awareness of what is actually going on in their own personal experience, because they've had the opportunity to see different family members? So there's a lot of complexity there, and that individual experience doesn't necessarily require genetic tests, but certainly we learn a lot from looking at the biology of populations as a whole.
Thanks. One of the really neat programs we do at the Jackson Laboratory is a high school-based program. We're in about 70% of the high schools in Maine, and we're teaching Maine high school students these concepts in genetics and genomics. And it's not just the science, it's the topics that Alyssa and our other panelists have just covered. Yes, you might have access to the information, but what can you do with it? What choices do you have with respect to your genetic information, or the genetic information that might be opened up to you if a family member receives a genetic test? So it's really a holistic approach about learning the science, but then also, how do you wrestle with it from a social point of view? So just a quick plug on that one. Great questions coming in. Let's see. Somebody asks, is there a way to witness this research in person or by video? Very fascinating, would love to learn more. So delighted this has sparked an interest in at least one person, so thank you for that. I will say that in my role, one of the things that I do is I welcome groups to the Jackson Laboratory on a very regular basis. We, for example, host a program of the Maine Development Foundation every year, Leadership Maine. Some of you may know about it. We host groups of teachers who come into the Jackson Laboratory through another program run by Maine nonprofit Educate Maine. We welcome chambers of commerce. Anyway, we don't have an open door policy because it is a laboratory, and there are restrictions that we have in place, as well as a working environment where fantastic hard questions can be asked and answered. But if somebody or a group, or you're part of a group that would like to have a focused on-site discussion about this, please reach out to me, and I can help put those pieces together. So I think we'll end because with one more question for each of you, and the hopeful sign is a lit, and that is one of the themes of today's session. So I'd like to ask, starting with Jake first, what makes you most hopeful? This is a question from the audience. What makes you most hopeful about the work you are doing, and do you foresee a future where genetic therapies could act as in a preventative way to combat addiction? And we'll just ask each panelist to share their hopeful thoughts for the next 5 to 8 years.
I am quite hopeful. I think there's a lot of opportunity for improvement, and that's where the hope comes from. I think that in the laboratory, we see a lot of these differences in animals, and it's a lot of, again, opportunity that we have in the face of adversity. Does that come in the form of, informed pharmacotherapy or genetic therapy? I think probably more on the pharmacotherapy side, at least in the near future. But no, I'm immensely hopeful.
Thanks. Jason?
Yeah, I think the biggest leap forward is the effect that biological research on addiction has had for helping to reduce stigma. So stigma of addiction and being a personal choice or a willpower issue is a big problem, and people need to realize that there is a biological basis for the way that things are occurring, and that there are interventions. There's medical assisted therapies, but there's just like a very high stigma. There's several medications that are out there for treatment of alcohol use disorder, but only 2% of people get medication for alcohol use disorder, whereas diabetes, 85% of the people get treatment for diabetes. So that stigma speaks to the disease and its lack of people getting it treated. So the more people understand, like my 85-year-old father finally gets it, I think that it's a biological thing, it's a disease, it's not somebody not going to church on Sunday.
I think for me, I'm learning so much more, and we all as a field are learning so much more about the interplay between sleep, exercise, life experience, adolescent drug use, and how that plays into future substance use disorders. And so the more we learn about mental health and addiction, and the more that we can come in at the ground floor and address those basic mental health and developmental issues, the better chance we have at reducing the impacts of a full-blown substance use disorder. And so I think that to me is the thing that we're learning the most. The way we've approached biological research in the past has really been to cut up the problem into lots of separate little pieces. And so in many cases, people would try to study mental health and substance use disorders in some sort of artificially purified way. And the more that we understand the interrelationships among these things, the better shot we have at modeling and intervening on the whole trajectory and potentially preventing people from getting to the state that becomes increasingly intractable.
Great. Thank you very much. I want to close just by offering to thank one individual and one group. The group is, of course, our panel. I want to thank you for your curiosity, your scientific drive, your kind of relentless asking of questions to get at these answers that we don't yet know. And I also want to thank you for always grounding the work that you do in the needs of the community and the needs of individual people. So thank you for that. Thank you for joining us today. The second thank you is for Gordon Smith. He's being thanked properly a lot today, and I don't know if he's in this room, but Gordon has personally been invested, curious, an advocate for the research of the Jackson Laboratory, including welcoming us to this panel on a recurring basis and giving us really a prominent stage for which to share the work that we do. So thank you, Gordon, for that. Thank the panel, and thank you all.